Swayam Prabha. Digital India. Educated India.
Welcome back to my lecture on applications of TALEN. So, part B. So, we have discussed about some of the promises and potentials of applications of TALEN and where it can be used apart from being cleaving DNA molecules, for example, as transposase, and then for activation and repression and DNA methylation and so on and so forth. So, let us continue our discussion with tail transposases. So, the designer DNA binding domain of tail can be used to guide a transposon to a specific region of a genome. Attempts were made to develop tail transposase by fusing the tail domain with a hyperactive variant of the piggyback transposase. The piggyback PB transposon effectively transposes between vectors and chromosomes using a cut and paste method. The resulting tail transposase enables the user defined genomic area in the human genome to retrieve a cargo DNA in a directed manner. The tail based targeting performed reasonably effectively with more than half of the insertions occurring within 250 base pairs of the tail target sequence and remained and the remainder insertion events occurring within 4000 base pairs. However, these approaches currently not practical because frequency of insertions is very low, 0.01% or to 0.014% for the stably transfected cells. But the potential of the technology can be displayed through this work. So, let us have a discussion on the tail recombinases. The tail DNA binding domain has been fused with site specific recombinases SSRs to achieve tailored genomic rearrangements. A hyperactive version of the catalytic domain from the DNA invertase GIN is N-terminally fused to a tail DNA binding domain to form a tail recombinases or tailors. So, you can see here the gene which is being fused to the N-terminal of the tail, and these tailors are usually utilized in pairs to carry out recombination. Apart from a DNA binding domain, the recombinase domain has predefined sequence dependent catalytic specificity. Therefore, a tailor pair requires at least partially conserved core recombinase motif of around 26 to 40 base pair located between the two target sites. Flip tail recombinases. Catalytic domain of the tyrosine recombinase flip can be fused with tail DNA binding domain to construct the flip tail recombinases. Like the tail gene recombinases, flip recognition target FRT or FRT like sequence consists of a degenerated 34 base pair motif, 4 to 5 base pair away from the two tail target sites, which is important for the activity of the flip tail. So, let us see the schematics of these flip tail recombinases. In E you can see the general mode of target binding of flip tail recombinases. In B you can see the flip tail and tail flip arrangement of the flip and tail modules and the respective possible relative arrangements of the flip and tail recognition sequences, and C is the schematic of the flip tail recombinases here. So, the flip molecule binds to the inner segments of the hybrid target sequence called the FRT-like sequence, while the tail module to the outer segments. The hybrid flip tail recombinases can be engineered by fusing the tail module either to the C or N terminus of the flip module, thus generating enzymes with the flip tail or tail flip architectures you can see in figure B. So, this flip tail you can see here the directionality, and this is the tail, and the flip is joined in this N terminus, and here it is joined in the C terminus. So, you have flip tail or tail flip, and this is a head to tail and this is a tail to tail. And then there is another kind of a arrangement here, you can see tail to tail, C terminus to N, and then head to head in this head to tail orientation in this figure number 4. The flip tail and tail flip recombinases can be engineered to bind to two possible arrangements of the inner and outer segments of the target sequences, head to tail and tail to head as shown in figure B, and they can be engineered either with a short linker that connects the flip and tail modules or with a long linker. However, short linker likely forms compact structure which is preferable. So, here you see this is the short linker and these are the long linkers. These are preferred over these because these linkers will add mass and volume, and the compact structures are much more preferred. So, we already spoke about this flip tail recombinase construct. Let us have some discussion on the tail based gene editors and regulators. So, here you can see the TALEN FOC1 hetero dimer, and this is a tail transcriptional activator, this is a tail transcriptional repressor. So, these FOC1 domain are different, and that is why they are hetero dimers, and this is a tail based transcriptional activator, and number C is the tail based transcriptional repressor. So, tail fusions with effector domains offer a broad range of applications like simple locus specific transcriptional activation and repression, and through direct induction of epigenetic changes on DNA or on histones, and using them for visualization and pull down of specific genomic loci. The application of tail with transcriptional regulations. Transcription activator like effector when fused with a functional domain instead of nucleus can regulate expression of endogenous genes. Transcription activators example BP16, BP64 or repressor domains such as the Kruppel associated box crab repressor domain can be fused in a vector by assembling tail DNA sequences with that of the transcription activator or repressor. In 2014, Zhang et al. Developed a multicolor panel of lentiviral tail crab expression vectors for knockdown of multiple gene targets. It was successful in knockdown of the 2 gene targets c-KIT and PU1 in bone marrows of recipient mice. c-KIT is a receptor tyrosine kinase and mainly expresses in hematopoietic stem cells and multipotential progenitors. PU1, also called SFPI1, is an ETS familial transcription factor and expressed in hematopoietic stem cells as well as multipotential progenitors. A multicolor panel of lentiviral tail crepe vectors constructed by Zhang et al. Replacing EGFP with the genes carrying cerulean, M-cherry iris blast or venous iris geosine expression cassettes was accomplished. The resulting vectors express 4 fluorescent proteins: EGFP, cerulean, McCherry and venous. In addition, PLV, tail crab, M-cherry blast and PLV tail crab venous geosine vectors were equipped with 2 drug resistant genes blast and geosine. So, this is the construction of a multicolor panel of lentiviral vectors which we have discussed, and these are the various fluorescent genes EGFP, ceru, McCherry and venous, and these last two McCherry and venous constructs are also given these drug resistance genes blast and iris and geomycin. And when transfected into cells, you can see the expression of these various fluorescence proteins. Let us look into the details of this experiment. So, in A you can see the maps of the tail crepe lentiviral vectors containing a multicolor panel of fluorescent proteins. Tail is fused to the N-terminus of the crab transcriptional repressor domain here. Individual fluorescent protein gene is fused in frame with crab via the T2A sequence. Drug resistant genes blast and geo are at the downstream of McCherry and venous fluorescent protein genes and separated by an iris internal ribosome entry site sequence. EF1 alpha human elongation factor 1 alpha promoter drives the tail crepe fluorescent protein genes and drug resistant genes. Blastic resistance to blasticidin as geo resistance to geosine, iris and internal ribosome entry site analysis is the nuclear localization signal, and these are the images of the 293 T cells transfected with individuals tail crepe expression vectors. Design of tail crepe transcriptional repressor for knockdown of c-KIT and PU1. A DNA binding domain was designed to bind the indicated target sequence within a proximal promoter region upstream of the transcriptional start of mouse c-KIT and PU1 genes respectively, and you have these construct already discussed earlier with crab and EF1 alpha, and this is the target gene and this is the transcription start site. The inhibitory function of the design tail crepe constructs was tested by generating 2 luciferase reporters for c-KIT and PU1 promoters respectively. The luciferase reporter assay revealed that both c-KIT tail crab and c-KIT tail to crab repress the luciferase activity of the c-KIT reporter more than 20-fold when compared to the control tail crab group. So, here in this diagram you can see luciferase reporters driven by the promoters of mouse c-KIT and PU1 genes on top and bottom respectively. The inhibitory function of the design tail crepe constructs were tested by generating two luciferase reporters for c-KIT and PU1 promoters respectively. Luciferase reporter assays revealed that both c-KIT tail1 crab and c-KIT tail2 crab repressed the luciferase activity of the c-KIT reporter more than 20-fold when compared to the control tail crab group. Tails have also been used to make fusion proteins which can be used for epigenomic modification. So, fusion of tail with enzymes which carry out epigenetic modification in an organism has been found to be quite promising. In epigenetic editing, the majority of the editing we have been discussing till now are genetic editing. We are now using these tail based approach for epigenetic editing. Here the DNA sequences of the gene are not changed. So, there is actually no any mutation, rather individual DNA molecules or histone proteins are modified with various chemical moieties to regulate gene expression. So, we have to know the difference between epigenetic editing and genetic editing. In genetic editing there is a change of the genetic sequence, but in epigenetic editing there is no any change of the genome genetic sequence, but some modification either in the base or in the histone protein takes place, and this can be done with tail based epigenomic modifiers, which are of two types, mostly based on the modifier proteins. Proteins that methylate or demethylate individual DNA nucleotides, and proteins that modify histones by adding or removing methylation, acetylation, ubiquitination or phosphorylation signals. Tails can be fused with any of these above epigenetic modifiers to carry out modification to regulate gene expression. So, here you can see some schematics of tail proteins used for epigenomic editing. So, first we need to identify the region of interest. This is the tail target sequence, and this is the area of interest. So, the tail portion will bind to the tail target sequence, and the modifier will act on the area of interest. So, you can see here some kind of modification being done on the DNA basis, and here modifications are being done on the histone proteins around which the DNA wraps. So, we can create tail targeted to regions of interest with attached effector. This is a tail protein and this is a TAT1. So, we can express the tail construct. So, these TAT effector removes DNA methylation. So, the tail binds to this DNA tail target, and these modifier TAT1 will remove these methyl group from here. Okay. So, this is being used for demethylation. So, other effectors like LSD1 can be used to make targeted histone modifications here. So, H3K27 is AC or H3K for ME12. So, these are in brief the concepts of epigenome editing. So, let us first discuss about the proteins that methylate or demethylate individual DNA nucleotides. Expression of target genes can be upregulated or increased or downregulated, decreased by using tail based epigenomic modifiers by directly changing the DNA's methylation state. 1011 translocation methyl cytosine dioxygenase 1 or TAT1, 1011 translocation 1 is fused with tail DNA binding domain, which causes demethylation of C at CPG sites, which we have shown in the figure earlier, and this results in subsequent upregulation of target genes. In contrast, fusion proteins comprised of a tail DNA binding domain and the DNA methyltransferases DNMT3A or DNMT3L or their catalytic domains causes methylation, the opposite of TAT1, of cytosine at target sites and thus decrease the target gene expression. So, by TAT1 we can upregulate gene expression, and using DNMT3A and DNMT3L we can downregulate gene regulation. The first demethylates, the second one just do the reverse, it methylates. The second type are the proteins that modify histones. The effector domains of 32 distinct histone modifying enzymes from various organisms have been investigated by Connerman and his associates, and they fused them to the DNA binding domain of tails. They found that several of them exhibited the intended transcription repressing impact on the target gene when their activity was compared in neuron cells. The histone methyltransferases KYP, TZSAT8 and NEU, and the histone deacetylases HDXC8, RPD3, C2A and SYN3A were found to be the most effective ones. A fusion to the lysine specific histone demethylase 1A LSD1 with tail carried out demethylation of histone without any examined off targets, which ultimately downregulated the expression of target genes in mammalian cells by removing enhancer associated chromatin modifications at their target region. Let us now discuss about one interesting topic where we can use chemicals to do base editing, and this is known as chemically induced proximity based editing or CIP based editing. CIP technologies have been integrated in a variety of ways to offer temporal controls in epigenomic editing and regulation. In the CIP system, a small molecule inducer promotes the homo-heterodimerization of two corresponding inducer binding adapter proteins that are individually fused to two proteins of interest. By controlling the proximity of POIs, proteins of interest, various downstream biological processes can be triggered upon the addition of the inducer. Many of these chemically induced proximity systems are readily reversible, and the induced dimerization or biological effects can be reversed upon the removal of the inducer from the system. So, the system is operational only in the presence of the inducer. If you remove the inducer, we can switch off the system, giving us total control. Lonjaric and his group exploited a feature of the designable transcription activator like effector DNA binding domain inducible by chemical inducer and a protease. So, these are called as chemically inducible tails, and here the heterodimerization approach is followed. So, there is a schematic representation of the heterodimerization approach. So, you can see here the tail bound to DMR A and DMR C bound to VP16 in the presence of some molecule like rapamycin here. They dimerize DMR A tail, DMR C VP16 will dimerize, and some effect will be there as a result of this dimerization. So, tail is fused to DMR A domain and VP16 is fused to DMR C domain. The tail domain can freely bind to the target DNA, and we know the mechanism of tail binding to specific DNA targets. And here the addition of rapamycin triggers interaction between DMR A and DMR C, leading to their dimerization. The VP16 fused with DMR C can recruit transcriptional activators to activate gene expression. So, these VP16 DMR C cannot itself bind to the DNA. We are using tail fused with DMR A, which binds to DNA and which allows the DMR-C bound to VP16 bound to DMR-C to form active partners which can switch on the gene expression. So, there is another approach, the reversibly locked approach, to do different kind of work, and these are also chemically inducible tails, and this is the schematic representation of the reversibly locked approach. Let us study the figure first. So, this is a Rapa tail, which means there is a tail domain, and you can see this with the VP16 bound here and some other components, and you see here DMR D, and then under such conditions these rapamycin bind to these domains, and it do not allow them to lock or bound to one another. And here you can see some other kind of binding orientation. Let us now try to understand which will switch on the gene and which will switch off the gene. This locked Rapa tail is composed of tail VP16 fused to homodimerization domains on both end. DMRD is here and DMRD is also here. The locked molecule is topologically inhibited from binding to DNA. Okay. In the presence of rapamycin, DMRD domains are inhibited from dimerizing, and the linear effector binds to the target DNA. The molecule is also unlocked by proteolysis with TVP, which removes the DMRD domain from one side, and now this molecule is able to bind to the DNA easily, because there is no any restrictions anymore. Let us look into covalently locked and uninducible tail VP16 direct 15 approach. The schematic representation of the covalently locked approach, the seq tail is composed of tail VP16 fused to intine fragments that cyclize the molecule through splicing. So, there is a splicing occurring here, due to which there is a cyclization. The locked molecule is topologically inhibited from binding to DNA. TVP can linearize these effector and allow DNA binding. and reporter gene expression, and here it is unable to bind and there is no any gene expression. So, in D you can see the schematic representation of the uninducible tail VP16 direct fusion approach. Apart from inducing tails with the help of chemicals, we can also induce them with the help of light. So, these are the light inducible epigenome editing approach. So, light inducible transcriptional effectors LIT lights were developed by Connerman et al. In 2013 to modulate the transcriptional dynamics and local epigenome landscapes of endogenous genes. Two proteins derived from Arabidopsis thaliana, the light sensitive cryptochrome 2 Cry2 and its interacting partners CIB1 dimerize upon photoirradiation at 466 nanometer. So, these Cry2 and CIB1 when exposed to light they will form dimers. So, you can see here CIB1 and Cry2 in the presence of light they are forming dimers, and here we are using tail to produce a tail Cry2 fusion protein, and this dimerization of CIB1 and Cry2 is exploited for epigenome editing. Cry2 and CIB1 are fused respectively to tail genome targeting module and VP64 or a variety of epigenome effectors including HDACs, methyl transferases, HET inhibitors as well as HDAC and HMT recruiting proteins. These epitails change the levels of H3K9 Me1, H4K20 Me3, H3K27 Me3 and several others and suppress the expression of GRM2 and NeuROC2 in primary neurons and Neuro2A cells in response to light irradiation. So, you can see in this figure that light induces the dimerization of Cry2 and CIB1 to direct epigenome modifiers to tail targeting sites for epigenome editing. TALEN has been widely used in many applications in disease biology like inactivation of hepatitis B virus replication in cultured cells, and this has the potential to be translated into therapies. Then also TALEN mediated gene correction in integration free beta thalassemia induced pluripotent stem cells, and for the application in biochemical production, example TALEN mediated targeted mutagenesis of caffeic acid O methyltransferase in highly polyploid sugar cane improves cell wall composition for production of bioethanol. So, in today's world where there is a lot of demand for bioethanol, TALEN is indeed a very promising technology. And others like transcription activator like effector nucleases used for metabolic engineering for enhanced fatty acid production in Saccharomyces cerevisiae. So, this can have a huge impact in the food industry as well as the fuel industry. So, before we end, let us make a comparison between the two technology platforms we have discussed so far. We discussed in both the cases their basic biology, and then we discussed how they are constructed or synthesized and what are the bioinformatic tools or softwares available to carry out their design or their optimized operations. So, TALEN and ZFN are mostly based on the FOC1 nuclease protein, and which is fused to a DNA binding protein domain, which is zinc finger in the case of ZFN, and which are tails in the case of TALEN. So, the difference of tails and ZFN is the protein DNA code they follow, which allows specific recognition between one tail and one nucleotide. ZFN based technology has disadvantages of the complexity and high cost of protein domain construction for each genome locus, and the probability of inaccurate cleavage of target DNA due to single nucleotide substitutions. TALEN can solve some of the problems associated with ZFN. However, the typical size for a cDNA encoding a TALEN is approximately 3 kb, whereas a cDNA encoding a ZFN is only approximately 1 kb. So, larger size makes TALEN harder to deliver and express a pair of TALENs into cells compared with ZFNs, and we have seen how large constructs with both the ZFN pairs can be loaded into a single vector and ensure its efficiency and efficacy. Now, let us discuss in brief a little bit about the two technology platforms which we have discussed so far, TALEN versus ZFN, and we know they are mostly based on the nuclease called FOC1 and a binding domain which is tail in the case of TALEN and zinc fingers in the case of ZFN, and both are proteins. So, these are mostly DNA binding proteins fused to a nuclease domain with which we can carry out genome editing. Briefly, the difference of tails and ZFN is the protein DNA code they follow, which allows specific recognition between one tail and one nucleotide. So, here in ZFN there are 3 to 4 nucleotides recognition by one zinc finger, and here one nucleotide by one RVD. The ZFN based technology has disadvantages of the complexity and high cost of protein domains construction for each genome locus, and the probability of inaccurate cleavage of target DNA due to single nucleotide substitutions. TALEN can solve some of the problems associated with ZFNs. However, the typical size for a cDNA encoding a TALEN is approximately 3 kb, whereas a cDNA encoding a ZFN is only approximately 1 kb. And we have discussed how both the left or forward or right or reverse TALEN ZFN can be loaded into a single vector, and their co-expression can be ensured to increase the efficiency and efficacy of the process. Larger size makes TALEN harder to deliver and express a pair of TALENs into cells compared to ZFNs. So, other things are as laid out in the field. You can see that the target sequence 2 into 12 nucleotide and more, this is little bit higher, 2 into 16, both are sensitive methylations, and then off target is quite high in ZFN, which is a disadvantage, and this is low in TALEN, which is a advantage in spite of it being hard to deliver due to its bigger size. And as already told, the cost is expensive in case of ZFN, and it is time consuming and difficult, and ZFN TALEN is relatively inexpensive and less time consuming. So, some of the references with which this lecture has been prepared. Thank you for your patient hearing.